All right, happy Friday, everybody. Good morning. Up here. Okay, so for those of you that just came in, please work on this Socrative entry quiz. And what we're going to do first in class today is we're going to finish up a tiny little bit about DNA replication that we didn't get to last class. Then we're going to get into the content for today, which is meiosis, which is what this morning's Socrative quiz is all about. But first, so we didn't quite get to the end of replication. What does this molecule, this double-stranded double helix, look like after replication's finished? So I didn't quite get to the end of the story. So can you imagine what it's going to look like if we draw the two fully replicated double helices? You draw it. I'll draw it too. But if there's a step 4, we've got 1, the original double helix, 2, the replication bubble has just been formed. The DNA strands are separating, replication is starting at step 2. Step 3, the chromosome is almost fully replicated. So what's step 4 going to look like? We'll have the 2 original black DNA strands. What else?
The 2 new strands. I don't think that it's supposed to come together.
So we'll have 2 new strands. Obviously, this is DNA replication. We have the separation of the 2 original strands. They each serve as templates to synthesize the next strands. I'll get you started. Those halves of those chromosomes look something like that. Blue, newly replicated DNA. Which strands are those? What do we call those? It starts with an L. It's either leading or lagging. Is it leading strand or lagging strand? Yeah, this turns out this is the leading strand. So the leading, it's okay. Thank you for being brave. The leading strand is the strand that gets replicated starting from an origin and keeps just going and going and going. So the long stretch of DNA there is the leading strand. What's the rest? Just say, yeah, but I'll get to you. What's the rest of this molecule going to look like? What am I going to fill in here where there's a blank? That second strand. In terms of colors, what's it going to look like? Right. So like the lagging strand looks like we're getting to over here, the lagging strand is synthesized from multiple red RNA primers that get put down by which enzyme? Primase, makes primers. Okay, so there's going to be lots of red primers. By the way, how did I screw up already? The leading strand started from what had to initiate synthesis of the leading strand? A primer. So there should be a little tiny bit of red at the start of each leading strand. Here. So then the rest of this will be primase has put down a number of red primers, and those initiate the synthesis of the rest of the chromosome. So this is what the last step might look like. You've got leading strand, the long piece of newly replicated DNA. You've got the lagging strands that are these short little pieces. Every newly synthesized strand, though, starts with red. Starts with an RNA primer. At this point, is the chromosome finished replicating? Trick question.
No, because the primers are RNA, not DNA, so they need to switch out the uracil for thymine, right?
Okay, so we've got 3 problems. One of them is we have RNA in our DNA. Waiter, what's this fly doing in my soup? What are the other 2 issues we have with the chromosome right now? It's not done replicating yet. Other issues? Something about binding? So why does this, for example, why does this top strand here not look like a chromosome? or a strand of a chromosome. Looking for the folks in the back for help. What's different about the upper strand here than the lower strand, besides the colors? How are they different? There's gaps. Right? What the hell's going on here? There's breaks in this DNA strand. It's not one continuous strand that goes all the way from left to right. So what's the solution to this? There's a protein. It's not polymerase in this case. Does anybody watch like 48 Hours or, I don't know, what are those procedural crime dramas these days? Help me out, folks. I don't watch TV. There's too many of let's do Grey's Anatomy. So you've got a corpse and somebody says, this victim has signatures of, or has ligature marks. What does that mean? Strangled. It's DNA ligase. Ligate to ligate. Those of you especially that are pre-med will need to know that that means to tie. You ligate. So DNA ligase. Ligature marks are also usually rope marks that somebody, if they've had their hands or feet bound or something like that, they get those marks on their arms or legs. Ligatures. Or strangulation too. So DNA ligase, Ligase, is an enzyme that comes in and fills in ligates, ties together those breaks in the backbone of the DNA molecule.
Oh, the one that creates the covalent bonds, right?
This creates covalent bonds between those. What are these little tiny pieces of DNA called? These are the Okazaki fragments. So ligase ties together or covalently binds the Okazaki fragments together. Okay. There's a third issue that we're not going to get into in detail, but that is that the ends of the chromosomes after replication don't completely replicate. So every generation, our chromosomes get a little bit shorter. Every cell generation, that is. Every time one of our chromosomes replicates, it gets a little bit shorter, and a little bit shorter, and a little bit shorter. By like 30 base pairs maybe, or 50 base pairs, or 100 base pairs. That's a big problem. So if we get a chance, we'll come back to how the cell solves its end replication problem later in the term. So 3 main problems at the end of replication. There's RNA in our DNA. What enzyme takes care of that issue? Something comes in and gets rid of the RNA. We have to replace RNA with DNA.
Polymerase 1.
This is a different DNA polymerase, and I don't require you to memorize the different polymerases and what they do, so don't worry about that. So it's with a DNA polymerase enzyme. So one of the many polymerase enzymes we have in our cells comes in and gets rid of, chews up all of those red RNA primers and sticks DNA in their place. So it's almost like I take my eraser and I erase. Oh, except I can't. I write over, how about that? Those primers with DNA. So that gets rid of the RNA primers, replaces with DNA. Thank you all for participating in the Socrative quiz. That's given me a good idea about what we need to cover today. So today's core concepts that I wanted you to get out of the reading and that we're going to practice a bit for the next 10 or 15 minutes or so are meiosis and gametogenesis. They're, of course, intimately related. Everybody hopefully appreciates this already. But this is where we're heading. So these are the 2 main things I'd like you to be able to do by the end of class. I want you to be able to understand why chromosomal non-disjunction happens and predict what its effects are going to be. We'll get into details. And then we were going to have a discussion about in vitro fertilization and genetic testing, which we started at the end of last class. Okay. So So first of all, what's the point of meiosis? Why do we need meiosis? Or what happens without it? What happens? Go ahead.
We're able to replicate or slash reproduce.
I don't know. I can envision a world where we could reproduce without meiosis. I mean, it's a made-up world. You want to join me in my made-up world for a second?
There'd be a lot of chromosomes. So keep adding.
Yeah. What happens if you're a sexually reproducing organism and you don't do meiosis? This is hypothetical. So let's say you're a sexually reproducing organism and you're diploid. There's your 2 copies of 1 chromosome during the interphase.
Your offspring would be tetraploid, right?
Right. So if you're a unicellular, exactly. So let's say you're a unicellular organism, like a yeast maybe, diploid. So pretend this is a yeast cell. And it comes along and here comes another yeast cell, it's diploid, and they undergo sexual reproduction. So let's say these are gametes and they fuse. What happens to their kids? They have a couple baby yeast. What are they going to look like in terms of their chromosomes? How many chromosomes do they inherit from dad in the upper left? 2. This is sperm and egg meet, essentially. So they get 2 chromosomes from dad. They get how many from mom? Okay, so siblings in the next generation are now what? They're not diploid anymore. Now you're tetraploid. Okay, no meiosis. So say these tetraploid yeast decide to inbreed. What happens to their kids if there's no meiosis? They're octoploid. They've got 8 sets of chromosomes. So what's the point of meiosis? What happens if we don't do meiosis? As the gentleman up front says, we get more and more chromosomes every generation, exponential growth in the amount of chromosomes an individual has. That's bad, people. Why is this bad? One of the main reasons this is bad is because the cell only has finite resources. How long is it going to take a cell to replicate 2 chromosomes? It'll take it twice as long to replicate 4 chromosomes, twice as long again or more to replicate 8 chromosomes, and so on. So at some point, there's an upper limit to the number of chromosomes a cell can tolerate because it can't replicate them fast enough to have them all replicated before the cell divides. And if the cell puts all of its energy into just replicating chromosomes, when is it going to do all the rest of the protein synthesis and things it needs to survive? So we do meiosis to maintain the number of chromosomes, to maintain our ploidy, so that we don't have this weird growth in the number of chromosomes. So meiosis and mitosis both start out looking very similar. And I want to walk through this again because this is a place where a lot of misconceptions arise. So I'm going to walk briefly through meiosis, compare it to mitosis. And when you have questions or concerns, please raise your hand. If I happen to be looking at this thing while I'm drawing, yell at me if you have a question, because I want to take care of questions as they arise. So just like our yeast, let's pretend that we're going to start with an organism that has 2 copies of 1 chromosome. So they're diploid. Here I'm calling blue, say, try that again. Blue is going to be chromosome from dad. Red will be a chromosome that was inherited at fertilization from mom. So it's a typical diploid cell. What has to happen first before meiosis? This is true of any cell division process. You're diploid. We have to temporarily become tetraploid. So what happens? Which part of the cell cycle does that? It's the last chapter we just talked about. Synthesis. Okay, so what does this look like after DNA synthesis? How do I draw these? How will you draw these chromosomes? How many copies of the chromosome from dad after synthesis? You start with 1. DNA replication happens. You better have, say it with me, people. How many? 2. Yeah, exactly. So we've got 2 copies of the chromosome from mom, and we've got 2 copies of the same chromosome from dad. So now let's say we're going to do meiosis. How many divisions are there in meiosis? Is there 1 cell division or 2? There are 2 division processes in meiosis. So here's the part that's always easy to look up on your tablets when you need to, but I still like you to remember. It's important to remember which, when this 1 cell divides, it's post-synthesis, post-S, it's going to go through meiosis. We're going to produce 2 daughter cells. How do I divide those 4 chromosomes into these 2 cells? Someone's got to tell me what to do. So you do. So there are 2 options. One is you do a chromosome from dad and a chromosome with mom in each daughter cell, 1 red and 1 blue. What's the other possibility? You put 2 blue in one, you put 2 red in the other.
Would you have to have like crossover?
This is meiosis. It turns out that in meiosis, the 2 copies that came from the same parent segregate together. They go together after the first division. And now I need to throw in a little bit of terminology that's really important. I've heard some of you use the term chromatid at some point in the past in this class. Here we have 4 sister chromatids. I want you to know that I really apologize on behalf of all of the geneticists in the world that did this to you. A chromatid is a double-stranded double helix. What do we call those usually? Chromosomes. So we have a second term for a chromosome. After DNA synthesis, in that period where we're temporarily tetraploid, they are no longer called chromosomes, they are called chromatids. I apologize. So these are 4 sister chromatids. Each one of those lines, as we've discussed before, represents a double helix. So you've got 4 copies of every chromosome, temporarily tetraploid. It's 4 sister chromatids. So in each of these first products, the first division of meiosis, we have the 2 sister chromatids that came from the mom, and in the other one we have the 2 sister chromatids that are the exact replicates of the chromosome that was inherited from dad. What happens next in meiosis? There's another division. So how many total products do we wind up making? 4. 2 cells divide in half, you make 4 cells. This should seem familiar. It's part of the question of today's entry quiz. So now how do we distribute the chromosomes? Yeah, 1 chromosome in every 1 cell. So you get a red, a blue, and a blue. The ploidy of these cells is haploid. They've got 1 copy of every chromosome. How is this different from mitosis? What would mitosis look like if we started with this same cell and did mitosis? divides in half. So this is where recombination, we're not talking about recombination yet, but you did bring up a good point that recombination does happen somewhere in this process. Do recombination later in the term. So what goes in these two cells? So which of each of the, so what pattern of lines am I going to draw here? Is it red, red in one and blue, blue in the other, like meiosis?
Blue, red.
You one of each. Because the point of mitosis is the products of mitosis are supposed to be genetically identical to each other. That's my one skin cell dividing into two skin cells. They need to be the same. The purpose of meiosis is to create haploid gametes, sperms. Did I say that? Sperm and eggs, sperms and eggs. Sperm and eggs that are haploid, so that when two gametes fuse, you restore a diploid organism. So which division of meiosis looks like mitosis? The first division or the second division? I hear both. I hear some firsts and some seconds. Explain your reasoning.
I think in the first one, because like you said, meiosis, we had the choice of choosing whether we wanted both of the chromosomes in one of the cells, like we wanted them to be mixed. And then mitosis, we get the mixed option, where in meiosis, we get the two from the same.
Right, so there's a difference here in the combination of the two sister chromatids you get in the products of the first division. In meiosis, it's the two from dad in one chromosome, the two from in one cell, the two from mom in the other cell. That's different than what happens in mitosis. Any questions about any of this? So mitosis, divide in half, every cell, the two daughter cells are genetically identical. Each gets one copy of mom's and one copy of dad's, chromosome 1 in this case. The product of meiosis is 4 haploid daughter cells. Now, relevant to in vitro fertilization, how many of, if you're a woman and your cell is undergoing meiosis, how many of these 4 meiotic products become an egg, an oocyte? That was one of the questions. There were a lot of different responses. This is really critical to appreciating genetics and fertilization. So guys, when one cell undergoes meiosis, how many sperm get made?
4.
Gentlemen, 4. Ladies, how many of those 4 products become primary oocytes or actual functional oocytes that can be fertilized? 1. Big difference between being a guy and being a lady. In fact, I don't know if you know, but this is how biologists determine who's a male and who's a female. It's not by what we look like on the outside, which is a good thing. It's the size of your gametes that matter. Females are the organisms that produce large, immobile gametes, like oocytes. And that's the reason that that oocyte production is asymmetrical. While these cell divisions are going on, 3 of the 4 of these, let's say, become polar bodies. They basically push all of their cytoplasm, all of the nutrient-rich goodies that are in there, messenger RNAs, proteins, and other things, into one of the cells, which becomes much, much, much bigger. So at the end of meiosis, one of these 4 gametes, or one of these 4 cells in women is really big compared to the other 3. That's the oocyte. In men, you get 4 sperm produced from every meiotic event. So that's the difference. Anybody who's a male produces small mobile gametes. Anyone who's a female is defined as such by producing large, immobile gametes. They stay put, the sperm come to the egg. Any questions or concerns at this point? So we've talked about one case in class of how trisomy causes a genetic disease. That's Down syndrome. There's another disorder that's caused by trisomy. If you have 3 copies of chromosome 18, you get Edwards syndrome. It's much rarer than Down syndrome. It's still pretty common though. 1 in 6,000 newborns or so is born with trisomy 18. You've got 3 copies of chromosome 18. So how does this happen? How do you wind up with an organism that has an extra copy of a chromosome? It has something to do with meiosis. So let's say we start back at that original concept of having a cell that undergoes synthesis. So you've got 4 copies of a chromosome. What can go wrong during meiosis that gives you trisomy? The sister chromatids don't separate, they don't split, they don't break up, I think you initially said, as they're supposed to. That's what's called non-disjunction. It's a double negative, non-disjunction. It means they junked. Come on, people, work with me. Non-disjunction. So let's say at this first division, one of the sister chromatids doesn't do what it's supposed to. What's supposed to happen here? Quick refresher. First division. Which pair of colors goes into the 2 daughter cells? So is this one of each color in each daughter cell, or reds in one and blues in the other? Reds in one, blues in the other. Okay. What if that doesn't actually work properly? What happens if that happens? The chromatids don't segregate the way they're supposed to.
Okay, well, aren't you talking about how at the end we're going to have haploid cells, so then they're going to combine, so that would be a tetrad instead of a...
So this is a triplet. I don't know what's going to happen. Let's see what happens, though. So you're exactly right. This is the cause of trisomies. What happens in the second division? Anytime there's non-disjunction, what are these 4 cells going to look like? What about the 2 on the left? What's going to be in one of the 2 cells? So in this division on the left, 1 and none. Right? There's only 1 chromosome. You're undergoing cell division. This is like 2nd grade math. 1 plus what equals 1? There's nothing in that second cell. No copy of this chromosome. Look on the right half. What are the possible ways that we split 3 objects up into 2 containers? What's going to happen? Right, we can't predict. There are 2 options, there are 2 possibilities. It could be 2 reds together in one and a blue in the other, or it could be a red and a blue in one and a red in the other. Regardless of which of those happens, we can't predict this. This is unpredictable at this point. You guess. That's the right answer. What happens if one of these, let's say these are sperm. They could just as easily be oocytes, but let's put some tails on them and call them sperm. What happens if sperm number 1 fertilizes a normal egg? You get a normal individual, or you get an individual with the wrong number of chromosomes? You get 1 chromosome in the sperm, 1 chromosome in the egg, you restore diploidy. What happens if you use a sperm that does not have this chromosome to fertilize a normal egg? How many copies of this chromosome do you have now? in the offspring? Just the one. What's that called? It's not trisomy, it's monosomy. Yeah, monomy. Whatever. You make up the terms, I'll go with them, that's fine. Monomy, it's monosomy, monosomy.
Is that where the case in one person will only have 23 chromosomes? Could that even be an individual? That even go up?
Well, I'm imagining the situation where we have a species that only has one chromosome. So imagine that this is, or you can imagine that this is a normal human situation. I'm just not drawing the other 22 chromosomes. For example, maybe this is chromosome 21 and this is how Down syndrome is created. Okay, thank you. I appreciate that clarification. So here's trisomy. What happens when this sperm fertilizes a normal oocyte? You've got 3 copies in the offspring of that one chromosome. So if you had, I don't really have time to get into that. Something interesting to think about perhaps. Every time one cell undergoes meiosis, if you get a sperm or an oocyte that produces an individual with trisomy 21, say, let's say this is Down syndrome, how frequently would you see an offspring produced that's monosomic for chromosome 21? Would it be more frequently, less frequently, or equally frequently? If I told you 1 in 10,000 births Down syndrome happens, what would be the frequency of monosomy chromosome 21? It'd be the same, because every time you create one of these sperm that's got an extra copy, you create a sperm that has no copy. It turns out that we don't see people that have monosomy 21 because it's lethal. You don't develop at all if you don't have 2 copies of chromosome 21. It turns out we can stand having an extra copy of 21. It causes some problems, but you're viable. You survive, you are born. We see people that have trisomy 21, but not monosomy 21.
Well, since for a human, you'd have each one of those lines represents 23 chromosomes.
Well, each one, yeah, so this is one of the 23, but the other 22 are invisible in this case. Yeah.
Because I was going to say, like, how come not everything gets replicated in just that?
So, what happens to the other chromosomes? Not just chromosome 21 or 18, the one that I showed in that previous screenshot, one of the individuals that has Edwards syndrome. What about trisomy chromosome 1? Do you think this happens to chromosome 1 as well? What do you think, yes or no? Can trisomy happen to any chromosome? Yeah. Do we ever have somebody go to the doctor and say, hey, I've got this weird thing going on in my life, check out my chromosomes? We never see people that have any of these other trisomies because they're all lethal. So if you've got 3 copies, there are only 2 chromosomes that we can survive with an extra copy of. That's 21 and 18. Any of the other chromosomes, if you've got an extra copy, or you don't have enough copies, it's lethal. You don't survive to development. It's embryonic lethal. So quickly, this is a question I asked you to think about when we, oh, go ahead, ma'am.
Can there also be trisomy of chromosome 13, 18, and 21?
There might be, okay, I will retract my comment. Until I investigate further, it might be that there are some other trisomies that are viable.
Yeah, because I just looked it up and it says it's called Patel syndrome.
This is why you should have computers in the classroom. I love playing this game called Prove the Professor Wrong, and this is a great example of it. So yeah, thank you. So I lied. I will retract my comment. There are a few other chromosomes, perhaps, that you can stand to be trisomic for, have 3 copies. It's the exception, not the rule. That's the bottom line. Thank you very much. So in part of the reading for today, the issue was, if you want to make a designer baby, or not, if you need to have help, assistance with reproduction for a number of reasons, how do you know which embryo, or which oocyte rather, to use for fertilization? If you're trying to pick an oocyte that has the right combination of mom's alleles to pass on to the next generation. Can you imagine what you do if you're working in a laboratory and somebody hands you their oocyte and says, here, check this and tell me which versions of my genes does this oocyte have? I want to know if I should use it to be fertilized. What would you do? Imagine, I know you're not in this situation, but imagine. They hand you their oocyte. What are you going to have to do to that oocyte to figure out something about its DNA? You have to you want to karyotype it, you want to do some PCR, amplify some DNA, sequence the DNA. To do that, you have to, who said that in the back? Take it out. You've got to take the DNA out of the oocyte and read the DNA. You can't do it while the DNA is still just sitting in the oocyte. So you disassemble the oocyte, you take out the DNA, you analyze it, and then you say, oh, shit. Sorry, this was the right oocyte. It had the right genetic makeup, but you can't use it anymore because it's broken. So how do you figure out which oocyte you want to actually use in vitro fertilization if you can't actually analyze the oocytes? It's all about the polar bodies. Those three other meiotic products that don't become the oocyte. Yeah.
I thought that even though during meiosis they end up getting different pieces of DNA, because we see the DNA in polar bodies, does it mean it's the DNA inside the oocyte because they have different DNA?
Exactly right. So how can we use the polar bodies to figure out what the genotype of the oocyte is? That's what this paper is all about. It's a proof of principle of an approach to figuring out, can we get the DNA out of the two polar bodies, 3 sets of chromosomes. So let me draw this again briefly so that we're on the same page. So if you have an oocyte that just has 1 chromosome, say chromosome 1, at the first division, you get the first polar body. That's the one that's shown on the top. It's got 2 copies of the chromosome, right? This is the first division of meiosis. And in the second division, you've got, I'm going to put an O in the middle of the oocyte, and on the left, the number 2, the second polar body. So you get 3 cells. You've got an oocyte, and you've got 2 polar bodies, essentially. So if you take any one of the polar bodies, I lied there too, but roll with it. The first polar body has how many copies of the chromosome we're interested in? 2. That's why they say the first polar body is 2C. It's got 2 times the number of chromosomes a haploid individual has. It's got 2C, where C is a chromosome. Okay. The second polar body only has 1 chromosome in it, so it's 1C. So what's on the y-axis? What's 0, 1, 2, 3, 4 mean? Number of chromosomes. How many chromosomes are at what's on the x-axis? This is why it's important to think about that when I asked last time. So it's 1, 2, 3, 4, 5, 6, 7, 8, blah, 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 22, and X. So this is all of the human chromosomes arranged left to right. So what does it mean? Here's the female pronucleus. That means the actual oocyte itself. What's the expected number of copies of a chromosome on every chromosome? What's the ploidy of an oocyte? Haploid, diploid? Haploid. 1 copy of every chromosome, and that's what we see. So when they actually destroy that oocyte, like I said, and sequence the DNA, they see there's 1 copy of every chromosome except for, wait a second, what happens over here on the right? How many copies of chromosome 16 are there in this oocyte? It's down here at 0. So they've taken an oocyte from a female human, they sequenced it, and they found that for chromosomes 1 through 15, there are one copy of each of those chromosomes in the oocyte, like there should be. Then they get to chromosome 16, there's no chromosome 16 in this, bless you, in this oocyte. Is this one you'd want to use for in vitro fertilization? Not if it's missing a chromosome. What else is wrong with this oocyte? It's got an extra copy of chromosome 19, and what else? It's missing chromosome 22. What does it mean? So if the oocyte is missing chromosome 22, where is it? It's going to be in one of the two other cells. It's going to be in one of the polar bodies. Does that make sense? So is that what they saw? Where was it? Chromosome 21, where's the extra copy? Here in the first polar body, where it should have 2 copies of every chromosome. So we've got 2 copies, 2 copies, 2 copies, and then all of a sudden you get to chromosome 21, and hey, there's an extra copy of chromosome 21 in that polar body. It's got 3, not 2. What do you take away from this, besides, holy crap, what am I looking at? When you look at all of this information, the sequence of one human oocyte.
You can use the polar bodies to determine what's in the oocyte.
Right, you've got 3 cells. If you know what's in 2 of them, you can figure out what's in the third. Because there are 4 copies of every chromosome. You just do addition. What's in the first polar body, what's in the second polar body? You subtract that from 4, and you figure out what's in the oocyte. But what's more important about this than we can figure out what's in an oocyte without actually sequencing the oocyte? What do these data actually tell you about meiosis in humans?
You can have like mutations, because you'll know if it's none of those other 2, then there won't be the.
Right, so we can do genetic counseling. We can predict which oocytes should be used if you want to produce an individual diploid for every chromosome. Is there anything surprising about this? What's shocking? There's something that's shocking to me anyway. I don't want to say it's shocking to you.
Shouldn't the first polar body have 2 copies of chromosome 18?
Right. And it's only got 1. So that must mean if it's supposed to have 2 copies and it's only got 1 copy there, where's the other copy? It could be in the second polar body. It wasn't in this case. It was in the oocyte. How many different chromosomes are affected in this one oocyte? 3? 3 whole chromosomes, plus, actually, if you look over here, part of chromosome 2, a little chunk, somehow. So one human oocyte, one mom produces 1 egg, could have been fertilized, is aneuploid or triploid. It doesn't have it, or it has too many of 3 different chromosomes in the same oocyte. I would have imagined it was just maybe 1 at a time. Like maybe it's really rare. And if you took any oocyte, if there was going to be a problem, it would be with 1 chromosome. They didn't just happen to find 1 oocyte that has a problem. We don't know now, really, I don't think, how frequent is this? How many oocytes an individual produces, or sperm for that matter, we haven't even started talking about sperm, might exhibit this. You have multiple issues all at the same time in the same cell. It could, maybe, not very likely to me, but I don't know. We have very little information about how these combinations of chromosomal issues are in actual living humans. Okay. So I want to set you up with another question. I love the questions that you're asking, and that's why we're not going to get through today what I thought we were going to get to. It's totally fine with me. But next class, we start talking about Mendel's laws of inheritance, some Punnett square work, and there's a Google Classroom assignment I set up at the start of class today. I was hoping we were going to have some in-class time to work on this, so now it'll be something that I'd like you to work on for next class. But I want to set up the story here. So this is the species I work on, so the last 5 minutes of class, I'm going to tell you the story. There will be a question to work on for next class. Not for points, just for you being ready to have useful conversations next class about this concept, Mendel's laws. These are diploid individuals, these worms that I work with, C. elegans. This is a hermaphrodite. So this one individual produces both sperm and eggs. So she can it can self-fertilize and produce its own offspring. This is one of these. I don't know how to pronounce this slash, but it's a she. Right, it's a hermaphrodite. So you can reproduce C. elegans in one of two ways. Either you can take one hermaphrodite, which you draw as the combination of both the male symbol and the female symbol, so that's one hermaphrodite, can produce her own offspring, babies, that are also hermaphrodites. Or you can take a male and mate him with a hermaphrodite, and they produce babies also. So there are two ways. This is very much like Mendel's peas. Peas can be selfed. You can mate a plant with itself, or you can cross a plant with another plant. So this is a good analogy for Mendel's peas. And I want to show you something really quickly. This is what C. elegans look like when they crawl. So they kind of have a snake-like movement. They make a sinusoidal wave, they crawl. Did someone say they're cute? That's awesome. Okay.
They're so tiny.
They're about 1 millimeter in size when they're adults. Now, this worm has an issue. It's probably hard to tell. You've got the link, so you can watch this on your tablets. Maybe you can zoom in a little bit more. This is a roller mutant. Instead of crawling on its side, it spins around and around like a corkscrew and turns in circles like this. It's unlike the original worm that just has a snake-like pattern. This one is a mutant.
Is it just stuck in a constant circle?
Yeah, basically. It's very sad. It is
What
sad.
kind of life is that?
No, it's a horrible life, and yeah, I'm a bad person for studying these worms. Okay, so it turns out that for this mutant, remember, these are diploid organisms. So if they have one mutant version, which I'm going to denote with a minus sign, this gene has a mutation in it, so minus. If you have one mutant version of the chromosome and one wild-type version, you're not a roller. You're normal. You have that snake-like motion. If you're going to be a roller like that worm we just looked at, it has to have both copies of that gene that's mutated have to be the bad mutant versions. Okay? That's roller. Now we've also got C. elegans that we've made transgenic. We've inserted a gene from the jellyfish that glows green. It's green fluorescent protein. And this is an example of the head of one of the worms that's been injected with this gene and its throat is green. So this is different from the roller because I'm going to use an asterisk to denote the gene that carries this protein, that encodes this protein, an asterisk. So if a worm has one copy, so an asterisk means it's the GFP gene. If there's only the GFP gene injected and stuck into one of the two chromosomes, the other chromosome is plus because it doesn't contain GFP. Or if both of the chromosomes contain this gene, either way it's green. So what I want you to do, you don't have to do this with a partner, although you can if you want to since we're out of time. Check out this worm. This is a green fluorescent roller. It's spinning around in a circle, and it's got GFP, a green fluorescent protein in it. So I was looking at this video and I was thinking, this would be kind of cool to have as jewelry or something you passed out at raves. Yeah, you like put it on your wrist and it rolls around on your wrist and it glows and okay. So here's the goal. Sorry, give me 40 more seconds. Your goal is to make green fluorescent roller worms like the one we see here. This is the goal because we want to go sell them at raves. Right? So here's what you've got. You have two different things that you can use. You've got some roller worms and you've got some GFP worms. We're talking about setting up crosses, Mendel's laws, etc. So please think about those three questions. If you had worms that were rollers and worms that were green, what would you do to combine those two traits together to make a green roller worm? Can you design a worm that has both green and is a roller that produces offspring that have both of those traits? And what's the best way to do that? So we're going to talk about this in class next time. Think about this as you are preparing for class and reading the paper, reading the textbook rather.